is to filter out what's in your blood. That's how you form urine, right? We form urine by filtering out substances that's in your blood. This slide shows you a higher magnification of the first part where that filtration happens. There are three steps in the formation of urine, and by formation of urine, we mean that that's the way that we get rid of waste products through our urine. So the three steps are: number one, glomerular filtration. By the way, I remember in my test on the renal system, I will have a picture of the nephron, maybe like this, or any kind, any any kidney picture with the glomerulus like this maybe, or uh, so I would use any picture, and I would I would ask, write down. I'm not going to write this down. I will say, write down the three steps in the formation of urine and use an arrow to point where in the kidney this happens. So the first is glomerular filtration, and then the second is called tubular reabsorption. And then the third is tubular secretion. Okay? It makes sense that this will be the first one, right? You need to filter out everything first out of this glomerulus. So this is you remember your simple squamous epithelium under the microscope? So what you, the slide that was prepared is really a slide of the glomerulus. And this is how it looks like under even higher magnification. So there's your glomerulus, these are your capillaries. And this is the first part of your kidneys. You see? It's a capsule. The best way to understand your kidneys is to think of this as a basin or the sink, right? The sink right there. You have the faucet. The faucet, water comes out of that one. And so you think of that one as the blood vessel. And then, right, the sink has, what do you call that? This, the thing that filters, the little thing that you put in there.
The screen?
The screen. Do you call it a sink screen?
No, I forgot what it was called.
A drain. A drain, right? So you have that. So this is the one. So you look at, and then you have these different tubules that will eventually end up in your PP. Okay? Eventually PP it out and go to the toilet. So there is your sink. See? And there's your glomerulus. Look at this part. Glomerular filtration, meaning you filter substances out of the glomerulus. So again, I'm going to write down what's flowing in my blood. I have my albumin, my globulin, fibrinogen, and then my RBC, white blood cells, platelets, and then my urea, which is the common waste, plus other toxic substances. I have water in there. I have my nutrients. By nutrients, you should know that I refer to your amino acids, fatty acids, your glucose, then whatever you have hormones here, right? Everything is here. So it's passing through, and what keeps, so what ends up here is just this one, the albumin, globulin, fibrinogen, RBC, white blood cells, platelets. All of this, as they pass through, they're gone. They entered my Bowman's capsule. Bowman's capsule is the first part of the kidney. Okay? This capsule, they just cut it, peel it open, the internal part, to show you how close they are. So I think the book uses the example of a balloon. If you have a balloon, think of a balloon, right? If this is a balloon and this is your hand, you squeeze that balloon, right? So what will end up is the balloon will be smushed like this maybe, and there's your hand. This is your hand, right? Okay, can you picture that? So that's the same as your Bowman's capsule, okay? So just think, and so it's so tight, the union or the attachment between these capillaries and your Bowman's capsule. This the simple squamous cells that make up your Bowman's capsule is referred to as your podocytes, the cells that line that are brushed against your glomerulus. They're only called that way because they have so many, a big space in between them. There, you see the podocytes. I think this is a nicer picture. You see, there's the podocytes of the Bowman's capsule, and there's your capillary. The red one is your capillary. This one is just showing you how really it's very easy for all of this to enter this capsule because they're so wide. The spaces in between are so wide. Yet they're not that wide to allow this albumin, globulin, fibrinogen, RBC, white blood cells, and platelets to escape. Don't forget that. Okay? So I'm going to go back to the first picture right here. So there. So this ended up here, and this ended up here. So I have my urea and my toxic substances. I have my water, vitamins maybe, my nutrients, my hormones, etc. They're going down there. First question, how do you think they ended up from here to here? What is the key for pushing them out? Remember, they don't have legs. What's the key is you have to have, what's the word? You have to have, how do you push it? What did you say? You have to have what?
Pressure.
Very
Pressure.
good. Pressure. Right? They don't have legs. They can't jump from here to here. It must be pressure that pushed them out. Now, do you understand why in the ER or in the hospital, when someone has low blood pressure, what is one of the first questions? What is the urine output? Right? If you have urine output is is a good gauge of the blood pressure of an individual. You have to have normal blood pressure to push these substances out, to filter. So glomerular filtration is the first step in formation of urine, which relies on normal blood pressure. Now, do you agree? Do you understand? Right? That's how this ended up here, because they don't have legs. They must have been pushed out. Okay? So, we normally vacillate between normal blood pressure, right? Sometimes we can have high blood pressure and also low blood pressure. They are varying conditions that normally happen to us within a 24-hour period. Do you agree? Sometimes your blood pressure will go down, sometimes it will go up. But the kidneys cannot afford to be affected by varying conditions of blood pressure. As long as you have healthy kidneys and healthy cardiovascular system, it doesn't matter. It shouldn't matter whether your blood pressure is so high, so low. When you get here, you have to have that normal pressure. Why? Can you predict? Can you tell me the consequence of, let's say, low blood pressure? If there's no way for your kidneys to adjust. I know that there are instances within 24 hours in me that I will have low blood pressure. Would it be the same maybe when you're sleeping, right? Your blood pressure goes down. Can you afford to have low? What do you think? What's the consequence if the pressure is low?
You can't push it out.
What did you say?
You can't push
You
all
can't
this
push
stuff
it out. That's right. You can't push out the waste products. right? Because pressure is the key to push it out. So if the pressure is low, you can't push it out. So what happens to this toxic stuff? It keeps flowing. It just keeps flowing. We can't afford that. Sometimes I know my blood pressure goes up. I know that. Especially when Baba drives me nuts, right? So, what will happen? Can you tell me what do you think can happen if the pressure is so high?
You can blow them out.
Oh, you can? Yes, very good. I can burst this, right? If the because remember, capillaries are just simple squamous, very easy, very very fragile, and if the pressure is high, poof, it will pop, and that's how you end up with maybe blood in the urine. But that's not the case with me. I don't see any. My urine analysis is still normal. So another thing that can happen with high blood pressure, if it's not that high to pump this out, maybe another thing that can happen is, do you follow? It can move fast. So when you push it too fast, you didn't give this enough time to filter out your waste. That's another thing that can happen with very high blood pressure, right? Not with high blood pressure. So fortunately, okay, God loves us so much that we auto-regulate. Your kidneys auto-regulate. I remember that was one answer to the question in your renal system test. Auto-regulation. Something to the effect of something like blood pressure. How do you explain? And the one word, just looking for one word, auto-regulation. The kidney, they say, according, I forgot now the name of the author, Holmes Human Anatomy, I think. It was written there that the up to blood pressures as high as, can you imagine? As high as 170 millimeters, 170, 120 over 80, even if it goes up to as high as 170, you will still form urine. Your kidney is able to auto-regulate. This is the key. This is the first step. When we get to step number 2, step number 3, that's easy. You need to first bring them out. Bring them to the kidneys. Then the kidneys will take care. Will be able to discern which one you want back, which one you want to throw out. But this is the key. So how does the kidney auto-regulate? Okay, that's the question. Auto-regulation. So there are 2 blood vessels here, the afferent arterial and the efferent arterial. Okay, and what is the wall? The wall of the artery. What is that key wall there? It has a thicker muscle, smooth muscle. Remember? So this one has no muscle, but this one has smooth muscle. So let's figure out. So you see that there's this JG cells, the macula densa. So these cells right here, and the macula densa of your tubules. So this is what's referred to as your JG apparatus. Okay? More than memorizing JG apparatus. Anyway, if you see this JG apparatus, it refers to these smooth muscle cells of your arterioles that are sensitive to blood pressure changes. So they're the ones that sense. If the blood that is flowing here is very high in pressure, to maintain filtration, can you predict? What do you think? How does the afferent arteriole respond? Will it constrict or dilate?
To slow down.
To slow down, because the pressure is so high. How do you think this afferent arteriole will respond? Will
It'll
it dilate?
constrict.
Very good. It will constrict. That's what you mean by autoregulation. So you have this series of cells here that are sensitive to, not series, but groups of cells. They're sensitive to changes in blood pressure. I'm predicting that when the blood pressure is high, It is the afferent arteriole that auto-regulates by constricting. So that when you get to the glomerulus, the pressure is still the same. Because if it's too much, you can burst this open, right? Or you can push them very quickly. There was not enough time to filter your waste out. How about if my blood pressure is low? Which one do you think will auto-regulate? The afferent or the efferent? Remember, blood vessels, they just respond by constricting. So which one will auto-regulate? Afferent or efferent? When my blood efferent, very good. If the blood pressure is low, the efferent will auto-regulate. It will constrict, vasoconstrict, right? So even if the pressure is low here, if vasoconstricts, it will still maintain that filtration pressure. So the key is to maintain a normal filtration pressure, which you're going to be asked to compute in your test again. Some of you missed that computation, so hopefully you will remember how to compute. It will come out in your test again. See, for 3 days now I've been having this. Like gas, not gas, you feel that there's something in there, like something here, yes, something like, there's like air in there, or water in there. I think I have gallstones. All of that super buffet now is okay, so look, don't forget this. A lot of you, well not, some of you weren't able to compute for filtration pressure. It's very important for us to know how to compute for filtration pressure. Remember when I asked you in your cardiovascular system, some of you didn't get that much. So look at, how do you arrive at, you need to be able to filter out. Your kidneys, all of us, all our kidneys need is 10 millimeters mercury of pressure. You can't go higher than that, you can't go lower than that. If it's higher, the nutrients keep passing, the waste keeps moving. It may be very fast, right? Or you risk popping this open, bursting. And if it's too low, maybe there will be no filtration. So you have to maintain that 10, that's enough for the kidneys to do its job. So again, you will see the words hydrostatic again, blood osmotic pressure, capsular. So when you say glomerular blood hydrostatic pressure, it's the pressure from the glomerulus pushing the substances out, right? That way. That is your glomerular hydrostatic pressure, isn't it? When you say capsular hydrostatic pressure, that must be the pressure from the capsule pushing it back in there. See, that's your number 2. And then what's your blood colloid osmotic pressure? Isn't that the one, the albumin right here, that's sucking it back in? See the arrow pointing back in? And then finally, you have your interstitial fluid. You have interstitial fluid osmotic pressure. I think I included this in your computation. So remember, the proteins here will suck it back there, right? So to compute,
Is that the capsular hydrostatic pressure?
the capsular hydrostatic is pushing from here to here. Hydrostatic is pushing it out, and then osmotic pressure is sucking it in. Okay? I know the reason why they didn't include that is because normally you shouldn't have proteins here. So that's why they didn't include this one. But if I included this in your test, I hope you remember. So osmotic pressure always sucks it in. Hydrostatic is always pushing it out.
So would you add that one to the GBHP?
Maybe, yeah, maybe.
I'm just trying to figure out where to put it in the formula.
Yes.
Because they don't have it.
Yes, you will add that. So it will be GBHP plus, very good, interstitial fluid osmotic pressure, minus Okay, minus these two.
Okay.
Okay. So you just use arrow, and that's what I do in the test. I will say this one for sure came out also. I will say using this glomerulus, please use arrows to point out these different values. So you're going to use arrows. And then my next will be compute for the net filtration pressure. Then the next question will be, do you think urine will be formed or not? This is the normal value for anyone. You should get to 10 millimeters mercury.
So if it's too high or too low, urine is not formed.
If it's too low, urine will not be formed, right? So that's why this has to auto-regulate to prevent that too low or too high. You got that? So we filtered out, we were able, this is a normal kidney, and it was able to filter out your nutrients. While the fluid was in here, did you notice what did we filter out? We filtered out the plasma part, right? Blood is made up of plasma and your cells. So we filtered out the plasma here. Once that plasma escaped your cardiovascular, it's no longer called plasma, it is called filtrate. So you know plasma, you know serum, they're all part of your cardiovascular system. You know tissue fluid, right? Tissue or interstitial fluid. You know lymph. You know CSF, of course. Now there's another term, filtrate. Filtrate is the fluid that's in your tubules, beginning with your Bowman's capsule, all the way until it gets to your pelvis. For our purposes, you cannot call it urine until it gets to the pelvis. That is the only time it is properly called urine for our purposes. Okay, right here in the pelvis. So all of the fluid that's in your tubules right here, the yellow structures, you call that filtrate. Do you understand how we filtered out? You understand pressure, right? Okay? You know how to compute for pressure differences. Now the next thing to compute for is step number 2, tubular reabsorption. Remember we said that most of the nutrients that have been filtered out right here, a lot of them I want back. I didn't mean to throw it out. I have to take them back. So now look at this. When you get to these tubules, very important, look at this one. You no longer have the help of your cardiovascular. Do you agree? You no longer have the help of pressure. No more, right? Because these are tubes. Blood vessel is separate. So as far as thinking, will blood pressure help me? Can I? No more. No more. The tubules now will have to rely on concentration gradient difference, right? How they will just move by diffusion from one area to the next. Are you following? Again, don't forget this part right here. This is not a blood vessel. So your blood pressure or albumin, they will not help as far as moving substances from here to here. The key to move substances between your tubules, so this is filtrate. Another term for the filtrate, do you agree? Tubular fluid. Tubular fluid. It's the fluid. The filtrate is nothing but the fluid in the tubules. So you can also call it tubular fluid. And the fluid here, within these spaces, is called interstitial fluid. Okay? Again, are you following so far? My cardiovascular system cannot help because this is not blood vessel. These are tubules. So blood pressure cannot help anymore. Once the nutrients are here, we have to rely on concentration differences because again, the fact is they don't have legs. They cannot move from one part to the next. There has to be, they follow the law of physics. There has to be concentration gradient difference. That's the only way they can move from one place to the next. Okay? And that problem is solved by their differences in permeability characteristics. Permeability characteristics. So this, can you follow this picture? This is your Bowman's capsule. This is the proximal convoluted tubule. And this is the descending loop of the nephron. This is the ascending loop. Then this will be another series, the distal convoluted, and this is the collecting duct that will empty into your pelvis. So if you look at this picture, what do you notice? Look at this picture. I see this is my descending limb or the descending loop. Look at what is able to move out here? Only water. So, and then here, do you see water coming out? No, just only salt. You see? They have different permeability characteristics. This part here, the descending limb, is permeable to water, but impermeable to salt. So can you predict what's going on? If this is permeable to water, but impermeable to salt, that means as they go down here, this becomes salty, 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 very salty. See, that's why, look at the concentration. It goes up from 400, 600. See, it became salt. Why? Because water is the only one that's able to move out. Then how does, what about what happens here? This is permeable to salt, but not to water. And you're able to do that because you're creating a salty environment. That's how water keeps coming out. Okay? And then if water keeps coming out, by the time you get up here, the salt here gets diluted. So salt will move down its concentration gradient. It's all just following a concentration gradient difference. And that is made possible by what? Permeability differences. Where this descending loop is permanent to what? Only water. And the ascending loop is permanent, ascending limb, sorry. The ascending limb is permeable to only salt. And that's how you're able to, did blood pressure play a role in? No, because this is not blood vessels. Let's look at how they move out. There's different ways to move substances in and out, move substances out of your tubules, okay? As far as movement of your tubules, of your fluids in your tubules, okay, so there's two ways, okay, in general. You have either they pass through here to the interstitial fluid. So this is your tubule. So this is the tubule of your kidneys. They can either, these chemicals can either move from here to here or right through the tubular cells. Remember that your tubules are made up of cells. So this type of reabsorption or transport is called transcellular reabsorption. This one is called paracellular, in between the cells. Okay? Again, you remember, we're going to go back to this part over here. Where is that? Over here, these are your tubules, okay? And your tubules are lined by your tubular cells. It's lined by epithelium. Simple cuboidal epithelium. And you're going to see them under the microscope. Okay? Simple cuboidal epithelium. And we said that the nutrients here, they're able to move from here to here. The key is permeability characteristics. So how are they able to move? We looked at the permeability characteristics. Okay, we know that now. Okay? We know that this is permeable to water, this is permeable to salt, and so they just follow concentration gradient. The next part is looking at how are they moved. They can either move from here, passing through in between the cells. You call that paracellular. Or they can be absorbed by the cell itself, and then from here it will be moved from the cell to the interstitial space. You call that transcellular reabsorption. Okay? That's how you move your substances. As far as water is concerned, there are two types of water reabsorption. One is called obligatory water reabsorption. And the other one is facultative. Facultative water reabsorption. This, what you see over here in the descending limb, this is called obligatory water reabsorption. They have no choice. They're gonna move because this is permeable. They pass through what you call aquaporins or water channels. And aquaporins are always present in the descending limb. So they're just obligated to move. Do you see this part right here? This is the collecting duct. This collecting duct normally have no aquaporins. So normally you won't see water being reabsorbed here. Okay? This is what what makes the difference between a concentrated PP and normal PP. When you need to conserve your urine, what is that hormone that's being secreted by your pituitary? Anti-diuretic hormone, yes, right? When you're dehydrated, right, your posterior pituitary will secrete that anti-diuretic hormone. And if you remember with the endocrine system, I asked you, what's the target organ of the anti-diuretic? And you wrote your kidneys. Now we're looking at it even in more detail. Where in the kidneys is the target of your anti-diuretic hormone? Here in the collecting ducts. These collecting ducts normally have no aquaporins. But in the presence of anti-diuretic hormone, the cells here, the tubular cells here, then respond by making aquaporins. What are aqua? For water. They are water channels. So that's how water is able to be reabsorbed, to return it back to your circulation. Do you follow? Do you understand this part right here? Again, don't forget, when it comes to the tubules, blood pressure has no role at all. The key to move the fluid in your tubules, this is not a blood vessel, this is a tubule, this is a pipe. The key to move them is you have to create a concentration gradient. And that concentration gradient is created by permeability differences, where this part permeable to water but impermeable to salt, and this part permeable to salt but impermeable to water. And then the next we looked at how do they move. They move either by transcellular or paracellular pathway. And then as far as water movement, you call that either obligatory water movement. This one is obligated, right? Because it's always open. Aquaporins are always present here. Whereas this is an example of facultative, because normally no aquaporins. How do they form aquaporins? Because antidiuretic hormone. When you have antidiuretic hormone, in cases of low blood volume, you want to conserve the water. So the antidiuretic hormone targets this one. It won't target this, right? Because it's always open already. It will target this to promote, to open up more aquaporins, reabsorb your water. So let's look at how that reabsorption of nutrients happen. That reabsorption of nutrients, okay? Oh, no. That reabsorption of nutrients, okay? I just want you to look at how we reabsorb sodium. Because all the other nutrients, see, look at, diffusion is the key, diffusion. There's no blood pressure involved in moving your nutrients from here to here. Look, very important for you to know, there are 3 compartments here, right? This is the tubular fluid. From the tubule, you have to move them from the tubule first to the interstitial area. And then from interstitial, they have to be moved then to your capillary, to your blood. So tubular fluid, and then interstitial or tissue fluid, or tissue, and then finally jump back in the blood. So they just move by diffusion. And as far as your nutrients are concerned, your glucose, your amino acids, your free fatty acids, they follow this glucose. They just move by diffusion. And you remember remember one of the characteristics of epithelium, it is polarity is one of the characteristics of the epithelium, right? Look at, as far as sodium is concerned, remember that sodium is always most abundant outside. Remember that? Sodium is most abundant outside, potassium most abundant inside. If sodium is abundant outside, you may wonder, how am I able to move it from here to here? It may not make sense. So here, very important for you to know your sodium-potassium pump, the AT phase. So there's a pump here. It requires energy to go against. Do you follow? Normally, sodium is always abundant outside of any cell. And that holds true for this environment right here. Sodium is very abundant here already. But how am I still able to pump it outside? Because it's okay, I understand how to move it from here to here. It becomes more concentrated here, it will just diffuse from here to here, right? How am I going to move it from here to here? That's the question. The sodium-potassium pump. Are you following, everybody? Does it make sense? Yes, no, tomorrow, something. You got it?
Yeah.
You got it?
Yeah.
Okay, good. The reason why I included this sodium potassium pump in the picture is to show you the importance of nutrition. That's ATP. Just to survive. Just the things that we take for granted, our heart pumping, our urine being formed, it needs energy already. That's why we have to eat. You see? Okay? Maybe I'll ask you, how, because you look at this one, no problem to move from here to here. Because in the tubule, your salts are so concentrated, diffusion, no problem. But how can that diffusion happen from here to the tissue fluid? That's the one that doesn't make sense. When sodium is most concentrated outside, it's going against. So that's energy driven, that AT phase. Okay? Okay. Once you get from here, how are you going to move it from here to here? Now, before we get to this part, I want to talk about transport maximum. Okay? Let's look at glucose. The one thing I hope you understand now, remember, we're talking about insulin. Remember, I said insulin is the key for letting glucose in. Remember that? So, in other words, your cell membrane also, just think like the bathroom, right? When you go to the bathroom and it's full, every cubicle is full, what do? We form a line outside. We have to wait until somebody is done using the bathroom. Do you follow? Okay? And that's the same thing here. Look at what happens. If there is too much glucose, there's a lot of glucose. This is going to your PP, isn't it? It's going to your PP, this is going back to your blood. If there is too much glucose, it is possible that all your glucose channels are filled and some of that glucose will spill in your urine because you filled it. It's called transport maximum. So what's that telling you? It's telling me that I took too much. I had too much sugar, which is not good. Remember, if you have a healthy kidneys, the presence of sugar always is a sign of abnormality. It's not normal. The kidney should be able to reabsorb everything. If it's overloaded, then that means it's not normal. Okay? In your test, I remember there was a section on urinalysis there. And you're going to answer that section in terms of telling me whether the kidney is working or not. We'll get to that one, but I want to finish this. I want to finish this last part first, okay? So we got to the part from tubular fluid. Now, I reabsorbed it and all my substances are now part of my interstitial fluid, my tissue fluid. Now, the next problem is how do I bring them back to my circulation, my peritubular capillaries, okay? So I will use this picture over here. So I got this part, the peritubular fluid. Now the nutrients and the way, no, just the nutrients are here, and the waste keeps passing through. Look at this picture. I like this picture. Look at, see the arrow. I mean the arrow for the water here, solid. This one broken lines to remind you that this is controlled by antidiuretic hormone, normally not present. So this is what you call facultative water absorption. This is, this is obligatory and this is facultative. Okay? So there's my afferent arteriole, glomerulus, efferent arteriole, and this is my peritubular capillaries. We revisit this efferent arteriole. Do you agree? By the time the efferent arteriole is flowing, the pressure here is way, way down. Do you agree? Right? So that by the time it branches into another group of capillaries right here called the peritubular capillaries, this one has no resistance at all. It's just going to receive, no problem. Pressure is down. Right? Plus, do you remember your albumin, right? Which keeps flowing, right? So your albumin, fibrinogen, globulin, your red blood cells, your white blood cells, and your platelets, they keep flowing in here. So this albumin plus low pressure here, don't you think it would be very easy to suck and attract all the nutrients back here, right? This is the key for exerting the anosmotic pull. And nothing will resist that pull because the pressure here is down. That's how you return your nutrients. Now you have clean blood. This is going to join your renal vein, go to your heart to be delivered to the rest of your body. Do you follow? And so this part right here, okay? When you get to this last part right here, this is made up of cells. Remember that everything is just made up of cells, right? This is simple squamous epithelium cells. This is simple cuboidal epithelium cells. So these cells right here is also made up, they're also metabolically active. So they're also producing waste. So the third part of formation of urine is called tubular secretion. I finally want to secrete everything that I want to get rid of, tubular secretion. And when you get to this part, which is your pelvis, it now becomes urine. So everything I have reabsorbed. This slide, I really like this slide. I just like it because I like the story that it's telling me. You understand? Very easy, isn't it? The thing you need to remember, right? Blood pressure is only a property of your cardiovascular system.
Where's tubular secretion? At the
Right
end?
here.
Oh.
So tubular secretion, you secrete into the tubules. Plus there's another one right here. What happened to that? This one. See, this is also another nice slide. So it's giving you, but the only thing about this one, it's not anatomically, so they uncoiled so that it's a straight line, but it's giving you a step-by-step glomerular filtration, tubular reabsorption, and then tubular secretion. Okay? It has to be a long process because you want to get rid of what you want to get rid of. This series of coiled tubes in your kidneys gives you a chance to reabsorb, to recapture what you didn't mean to throw out. That's why it's a long coil. Okay? So for the kidney to do its job, what is the product of kidney? The kidney's doing its job. What do you call the product? It's called. What is the product? Oh.
Metabolic waste or urine.
Urine. Very good. It's urine. Okay. It contains your metabolic waste. So, how do you evaluate kidney function? The most basic way to evaluate kidney function is by urinalysis. So what does urinalysis monitor or evaluate? The function of your kidneys. Slide number 71. The function of your kidneys. Okay? Okay. There is a section in your test that will ask you to tell me whether this is reflecting, it's a normal or abnormal. Write down where if this is a normal kidney function or abnormal kidney function in a urinalysis. Okay. So look at, is it normal to find sugar in the urine? No.
No.
First of all, dark PP, light colored PP is normal. As far as the kidneys is concerned, do you understand? That is telling me that my kidney is working. Yes, I know I have dark PP when I'm dehydrated. But as far as my kidney is concerned, my kidney is working. So it's a normal kidney function. It's normal for my kidney to conserve PP when I'm dehydrated. Do you understand? Those are two separate things. So in the test, you will answer in terms of, is my kidney healthy or not? This one, if this is present, that means my kidney is no longer working. So that's abnormal. Do you understand? So let's say if I write down, I think the test bank even says rusty colored urine. That's normal. It's normal because my kidney is working. That's just different darkness of the urine. Okay? It's not normal if you find glucose and proteins. They should never be in your urine, especially the proteins, right? In the first place, protein, if we trace the flow of protein, they should have never jumped out of the cardiovascular at any point. Glucose was able to jump out because it's very easy, but this one, no. Okay, so this should not be normal. Is it normal to find calcium in your urine? Yes. Urea? Yes. Your phosphates, all of these salts? Yes. If there's too much, this means it's telling me again what? My kidney is working. If this is present, no more. Maybe my blood vessels are broken. So that's not normal. You need to know these terms. Normal blood volume is about 1 to 2 liters a day. Polyuria, if I just ask you to define polyuria, you can write more than 2 liters a day, or you can just write increased urine output. That's okay. Oliguria is decreased urine output or less than 500 a day. Anuria is no output. 0 to 100 is considered anuria. Okay? And then, what about if there's white blood cells in your urine? That's called pyuria. So you should be familiar with these terms also. Hematuria, pyuria, ketonuria, proteinuria. Okay? Another way to test for kidney function, do you agree? In addition to urinalysis, a blood test can help in assessing kidney function. Do you agree? Because isn't the job of the kidneys to get rid of waste that's in your blood? So if the waste is still in the blood, that's telling me my kidney is not working. Have you heard of BUN? Blood urea nitrogen. Urea is the main waste product that you want to get rid of. That's the number one toxic waste product. So if it's in the blood, if you have high BUN test, isn't that going to tell me that the kidney is failing? Right? There's other, like your inulin clearance or creatinine clearance. Have you heard of creatinine clearance? Mm-hmm. Clearance means how Clearance. Where's clearance? Clearance means how your kidney is very effective in clearing a certain substance out of the body. So they do creatinine, they inject you with a certain amount of creatinine, and then they measure how much of it was released in your kidneys. If there's a discrepancy, that's the creatinine clearance. You should have been able to clear that. If there's a discrepancy, then you know that your kidney was not able to normally clear it out. Do you understand that you can assess kidney function through urinalysis and blood test? You understand? Okay? So now that we have formed our urine, let's look at how we get rid of it. We're done. We're almost done. Okay? How do we get rid of it? The next organ after your kidneys is your ureters. Don't forget the what we just talked about, all the functions, urine formation, etc., filtering out, is carried out by your kidneys only. The ureters, the bladder, the urethra, they have nothing to do with the job of your renal system. The ureter. What does the ureter do? It just transports the urine from your kidneys right here to your urinary bladder. So I am looking at the anatomy of my ureters. What do you think will help your ureters in moving the urine from here to here? One thing obvious is, yes, very good. Don't you think so? Gravity. See? That's why it's very important. See, look at the normal anatomy. If you kink that urine, you compromise the formation, the transportation. You want to get rid of your trash. That's your trash. This is your garbage collector, right? So I know gravity, number one. You remember peristalsis? Okay? That also helps. And also hydrostatic pressure. Right? Like a waterfall, the hydrostatic pressure from all of that fluid building up, you have no choice but by pressure you fall down like a waterfall. So those three factors help in the transport of urine from here to here. Gravity, peristalsis, hydrostatic pressure. Now we get to the urinary bladder. Okay? Let's open up this bladder right here. What do you notice? Again, rugae. So when you open up your bladder, which you're going to do next week, not your bladder, but the kidney, the cat's bladder, okay? Rugae again, temporary folds. So the bladder should be a holding chamber until I'm ready to pee-pee, right? Some people, they want to hold their pee-pee because the rest area is stinky. So what makes the bladder an ideal holding chamber? Number one, rugae. Number two, do you remember? Transitional epithelium, right?
Stretches.
Yes, very good. Transitional epithelium. Number three, the smooth muscle. Isn't that a property of smooth muscle? It is able to stretch. So these features make the urinary bladder an ideal holding chamber. I want you, when you open up the bladder, to look for this. There's an area in the bladder called the trigone. Okay? It's a triangular shaped area that is marked by three openings. Two from the ureter and one to the urethra. So the urethra is nothing but an area where there's a constriction from the bladder. By the way, what makes the bladder ideal for holding is this one. But what makes it very efficient in pushing out is the thickness of this smooth muscle. It's very strong. They call it the detrusor muscle. It's 4 layers of smooth muscle. So very strong in emptying it. Detrusor muscle. Look at this ureteral openings. Do you notice it's not even called a valve? It's just called ureteral opening. The reason why it's just called a ureteral opening is because just like your valves in your veins, they are see, it is a physiological valve only. It is not top connective tissue. So what is, it's just a modified fold of your epithelium again. What is this telling me? It's telling me that it's very easy. Look at this. There's no chordae tendineae that's anchored. None. There's none. It's very easy. If we hold our pee-pee, do you agree? If we hold our pee-pee for a long time, do you think it's very easy for this to pop? Evert, go back up there. Very easy, isn't it? Because it's not an anatomical valve. It's not like the valves in my heart where I have the chordae tendineae preventing the valves from everting or prolapsing. Nothing like that. This is anatomically explaining why holding your pee-pee is not a good thing. It cannot, it doesn't have that property of preventing backflow. It doesn't have it. Okay? So, can you imagine what we do? What's here is garbage. And this is very close to the openings right here. So it's very possible, especially with females. With males, they have a longer urethra, but females have a shorter urethra, closer to the bacteria here. So this garbage is nice bedding, board and lodging for the bacteria here. If we don't get rid of this one, it's very easy for the bacteria to poof, enter, cause infection, poof, go up, right? Why? Because it's not anatomical. Okay? I remember asking this in the test, something like that. It's just a physiological valve. They don't even call it valve. They call it opening. Okay?
Is that why you end up with a kidney infection?
Correct.
If you don't correct the urinary tract infection?
That's now, see? Now you understand, right? It's all just anatomically explained. Now we go to the urethra. This is the last part that helps us to get rid, right? So the urethra is guarded by sphincters, two sphincters. An internal urethral sphincter, and the internal urethral sphincter is nothing but the base of the bladder. Right here you see, it's the area of the urinary bladder, the smooth muscle that has increased muscle tone. So the internal urethral sphincter is smooth muscle. And the external urethral sphincter, which is here, is skeletal muscle. So how do you urinate? Called micturition. If I asked you to define micturition, you're going to write urination. Defecation is, what's defecation?
Pooping.
Pooping. Micturition is urinating. So again, you have two sphincters there. And by the way, I think it is so funny for the little boys when they feel like going to the bathroom, okay? I want you to look at the urethra in females and in males. So there is the internal urethral sphincter. That's just the end of the bladder. That's where the internal urethral sphincter is. It's what we call it, it's smooth muscle. The external urethral sphincter is right here at the perineum area, okay? It's the same for males too. I think it's so funny how the males try to squeeze this opening, trying to prevent the pee-pee. No, the external sphincter is right here. It's at the perineum, okay? But they have a longer urethra than us. So females, very easy. Look at the bacteria, very easy to go up there. And holding your pee-pee, knowing that that's not an anatomical valve, we are in danger, okay? So let's look at this micturition reflex. when our bladder gets filled to about 400 mils, that's when we start feeling the urge. So remember, you stretch your bladder, it triggers a reflex which involves your sacral spinal cord. And the result is relaxation of the internal urethral sphincter. That's when you feel going to the bathroom because your internal sphincter is relaxed. But your external is controlled by you. It's voluntary, isn't it? It's skeletal muscle. So you go to the bathroom, and what do you do when you sit in the toilet? Don't you go again, right? Or some people, you squeeze your belly so you empty it. It's pressure. So you have to increase that abdominal pressure to squeeze all the urine out. And then finally, you flush the toilet and it's in your puget sac. So that's the end of the urinary system. Okay? Very easy, isn't it?
Yeah.
So don't forget your other homework